Hello everyone, my name is Carly Bothman. I am a student at Eastern Illinois University, and this is my presentation on the effect of high maternal folic acid intake on offspring's health, a systematic review. We will begin with a brief introduction. Listed here are the four main topics that I will be touching base on for the background information. This first little box here stating different forms of folate/folic acid. I'm not going to go into a whole lot of detail there just because as dietetic students I would assume we all know the difference there, as folate is that natural food source form and folic acid is the synthetic form that's used for fortification purposes and supplements. So when we think of the importance of folic acid, I think the first thing that comes to mind for a lot of us is going to be the prevention of neural tube defects. That definitely is a huge one. But backing up real quickly, several human and animal studies alike have consistently shown the essential role folic acid plays in the development of the brain and nervous system. So that can kind of feed into that idea that it helps with neural tube defects just because of the brain and nervous system being associated with that process. However, wanted to note that the specific mechanism by which folic acid plays a role in preventing neural tube defects is still iffy. They have some ideas and inklings, and some of these other bullets hint to them, but as far as a concrete mechanism, that is still being studied and is unknown. But specifically, as far as studies go, there was a series of randomized clinical trials in the early 1990s that showed periconceptual folic acid supplementation prevented 50 to 70% of neural tube defects. So when I say periconceptual folic acid supplementation intake, that means that time before conception, so leading up to pregnancy. Folic acid has also been found to produce immunological effects, meaning it could influence the development of allergic diseases. And finally, folic acid has been shown to play a role in a wide range of physiologic processes surrounding DNA. Specifically, the synthesis of, the repair of, and the methylation of DNA. And for those of you who might not know what methylation refers to, that's just that transcription process of genetic transcription of the DNA. So basically turning on and off certain genes and kind of differentiating the functions of the genes. And then folates are also going to be essential for the synthesis of critical compounds needed for cell division. And it has also been seen that folate can play a role in epigenetic regulation of gene expression. So all of these things allude to the importance of folic acid, and several of the studies in the systematic review will actually come back and relate to these importances. So back in the 90s, when they were starting to realize that there was a need for mandatory fortification purposes for the prevention of neural tube defects, this really stemmed from the fact that about half of all pregnancies at that time were planned. So that meant that a woman could definitely be denying her child of necessary folic acid intake during the most critical period of folate demand, which is those first few weeks of pregnancy. And so that is how the mandatory fortification of folic acid of grain products in the U.S. was implemented in 1998. Specifically, it is stated that 140 micrograms of folic acid should be added per 100 grams of grain. Then after this mandatory fortification was implemented, it was observed that serum folate levels were increasing, and this was specifically seen in a sample population from the U.S. Additionally, this mandatory fortification was successful in reducing the prevalence of neural tube defects, so this has been seen in a lot of studies. And then by 2009, 52 countries had decided to adopt mandatory folic acid fortification regulations. And then in Europe, what they typically do is they just allow voluntary fortification, meaning it's up to the manufacturers to decide if they would like to fortify their products or not. This final slide on background information is really going to feed into the purpose of the systematic review, as there has been an increase in maternal intake of folic acid observed over the years. So real quick, want to note that an intake greater than or equal to 1,000 micrograms per day is going to be coinciding with that tolerable upper intake level that has been set by the Institute of Medicine. And what we had seen is several animal studies which have drawn attention to a concern of folic acid supplementation in dosages at or above that intake level, which we will see some of those here shortly, but just basically studies showing that there are associated risks with this, and therefore we need to start looking into it. And along with that, several studies have alerted to the fact that folic acid intake is exceeding the upper level among pregnant women. So we saw that there's associated risks in the animal studies, but now we're noting that pregnant women actually are getting to those levels. So now we need to combine the two and see if we can do any human studies to see if there's correlations and what those associated risks might be. And then the reason or the cause behind this excessive folic acid intake is more so associated with the fact that there have been added levels from fortified foods. Physicians are prescribing prescriptions with folic acid in them, and also women are always encouraged to use an over-the-counter prenatal vitamin when they are pregnant. So all of these things are leading to an increased maternal intake of folic acid, and now we need to see what that might mean for the health of the offspring. So with all that being said, the purpose of this presentation is to reveal the findings from a systematic review that targets the question, does maternal oversupplementation of folic acid cause adverse health problems in offspring? Real quick, I want to note that the term oversupplementation for the purpose of this study is going to refer to any intake that is greater than the current recommended level, which sits at 400 micrograms per day. So anything above that 400 level is going to be considered oversupplementation. So now we will transition into the literature review process. So again, the objective was to assess the effects of high maternal intake of folic acid on the health of resulting offspring. So to do this, I utilized two databases. I used the CINAHL, I call that CNAL, I don't know if that's how you would really say it. So I used that and PubMed. And to touch on my inclusion criteria, I separated it based on animal studies versus human studies. Obviously, there's going to be some differences there. So starting with the animal studies, obviously I wanted to look at pregnant mothers and specifically their folic acid intake. With these animal studies, I wanted to be sure that there was a controlled diet compared to a high intake diet, so high folic acid intake diet. I wanted to be sure that the effects were evaluated among the offspring more so than the mothers, and then I did want it published within the past 10 years. The inclusion criteria for the human studies are similar, so I also want to be sure that the offspring were the ones being evaluated the most. Needed to include pregnant women, especially considering their folic acid intake. The main thing with the human studies is I wanted to be sure that they considered both the folic acid intake through supplementation and diet. So both of those together, and again, that time frame was utilized. So for the design, as far as my study selection went, I utilized Zotero, and what I first did is I had it remove duplicate articles. Then I first screened the titles, and I eliminated some articles just simply based on the titles. I could tell right away that they were not going to give me what I wanted. And then I moved on and I further excluded articles that had abstracts that for sure pointed out the fact that they were not going to answer my research question. So ultimately, like I said, articles were removed. And then I noted there's some of the keywords that I used for my searches. Things like folic acid, pregnancy, maternal folic acid, offspring, and high folic acid. So these are the results of the study selection process as presented in a Prisma diagram. So again, using both databases, the CINAHL Plus and PubMed, I was able to accumulate 196 articles. For both databases, I used the search phrase high folic acid and pregnancy. This was determined based off of trial and error, just to see what would give me the most relevant sources. So again, 196. I did not utilize any other outside sources besides those databases, so that was why there's a 0 there. I at least didn't utilize any for my primary study selection. Again, Zotero was used for removing duplicates, and that narrowed it down to 173. And from there, 97 records were screened. And the initial screening process, like I mentioned previously, I quickly went through the titles and was able to see if those key terms were there. If they were not, I excluded them and tossed them out just because I was pretty aware of what I was after, and a lot of the titles gave me a big clue that they weren't going to help me out. So that's how 58 records got excluded. And then from there, with the leftover, 30-something articles, I looked at their abstracts and specifically was paying attention to making sure that the inclusion criteria were there. So that left me with 21 full-text articles to assess. And with those, I really honed in on that methodology, again, to make sure that the inclusion criteria were there. And there's a few other reasons that some articles got excluded, but in total, 11 full-text articles got excluded. So that brought me to 10 studies for the purpose of this systematic review. To touch on why some of those articles were excluded, I came up with two main reasons. First and foremost, just many of them did not meet the inclusion criteria, which obviously was a no-no. And this was more relevant to the human studies. The fact being that I really wanted to evaluate the overall folic acid intake in the maternal diet, not just the intake through food sources, but also through supplements. And so what I was noticing in a lot of human studies is that they were only evaluating one or the other, and it was very important that the studies were evaluating both in combination with each other. And then the second reason for discarding articles was the fact that just upon further reading, some of them were not relevant. And this was more pertinent to the animal studies. Just because animal studies are able to control so many more extraneous variables and their methodology is a lot more complex, their evaluation measures just weren't measuring up with the human studies. They were just a little too far out there and complicated, so I had to kind of reel it in and make sure that the animal studies were going to be somewhat comparable to the human studies. As previously mentioned, I was able to produce 10 primary studies for the purpose of the systematic review. And this slide is just touching on the differences between my studies, and there were many, mainly because I had to utilize both animal and human studies. So there were going to definitely be differences in the characteristics of those participants or the subjects of the studies. As far as the total number of participants, the age range, and the offspring evaluated, and then species were also going to differentiate. The methods also varied quite a bit between the animal and human studies. Basically, what you'll see in the human studies is that both the amount of dietary folic acid intake and supplementation intake will be considered and was addressed in all the human studies. For the animal studies, though, a common trend for the methodology was that they had a control group, fed a controlled diet, and then they also had a high folic acid diet given to the other group. So I was able to really compare the two. And then, of course, there were variations in the outcome measures by which all of the studies, almost all of them, were looking at a different health outcome. This table is a good example of the general format I used for my data extraction. So in the next several slides, please pay special attention to the text formatted in different colors, as that information is what's going to be most differentiated across the studies. And also you'll see highlighted text in these upcoming slides, and that's simply just to draw your attention to the main results of the studies. And I'm saying all of this now because I'm only going to be briefly mentioning each individual study. I'm not going to go into a whole lot of detail, so feel free to pause this presentation if you need to spend a little more time reflecting on the information on those slides. Starting with the human studies, briefly, I'm just going to mention overall what to look for in the human studies. So all of them are going to have a very similar intervention design where they utilize a food frequency questionnaire as well as a folic acid supplement questionnaire to gain a generalized idea of just how much folic acid intake is being had by the mothers. The main differences are going to be the age at which the offspring are evaluated, the point at which these questionnaires were administered, and then the evaluation measures. So looking at this one, this study specifically was looking at the association between the maternal folic acid intake and the risk of asthma and or atopy. And overall, what this study revealed is that maternal folic acid intake greater than 578 micrograms per day was associated with an increased risk of offspring asthma. This next human study was evaluating the risk of cow's milk allergy prevalence in children in association to the maternal folic acid intake. So overall, what this study alludes to is the possibility that high maternal intake of folic acid may be associated with an increased risk of the offspring developing cow's milk allergy. So again, going back to some of that background information, I talked about the epigenetic factors that folic acid can have on gene expression. This would definitely tie into that, as allergic responses are a part of that epigenetic idea, I guess you could say. So this study does make sense as to why a higher intake of folic acid might lead to a cow's milk allergy in the offspring. This is one of the studies that I had sent out the full text article to you guys in advance. This one was looking at the association of the maternal folic acid intake with a score on the MSCA test, so the McCarthy Scales of Children's Abilities test. Basically what this test is looking at is the overall cognitive ability of the offspring. So overall, what was seen is that folic acid supplement dosages at the tolerable upper intake level during periconception may be associated with lower cognitive ability of the offspring. One difference I wanted to point out about this study in particular is that its location is in the US, meaning that it was done in a country where mandatory folic acid fortification takes place. The past 3 studies were in countries that do not require mandatory fortification. So this is something to note, because as we see in the overall findings, that first bullet, it says the mean intake in the first trimester was 930 micrograms per day. So that's a lot higher than the mean intake seen in the past 3 studies. So I just wanted to point that out real quickly. But overall, what this study looked at is that higher maternal folic acid intake was not associated with an increased risk for the development of asthma in the offspring. So this is the first study to actually have a positive correlation of high folic acid intake. They saw that it reduced the incidence of asthma. So something to consider there. Out of all of the human studies, this one had the most differences, I guess you could say, as far as the methodology. So it was in the US again, so probably got to take into consideration that mandatory fortification regulation. But how this one was set up with the participants is that it was still mothers and their offspring, but how they divided them up is they looked at offspring who had typical development versus those with an autism spectrum disorder versus those with a developmental delay. So they separated the offspring into those 3 categories based on wherever they fell, and then reflected on their mother's folic acid intake during pregnancy to see if there was an association there between where the offspring fell among the 3 groups and the amount of folic acid that the mother was taking in through her diet. Another thing of significance here is the intervention design was quite different from the last ones. So it still took into consideration both dietary folic acid intake as well as the intake through supplements. However, this one was a little more limited as far as how much information was obtained. It only accounts for some of that dietary maternal folate intake because it was not an extensive food frequency questionnaire like the other studies had. But overall, what was found is that higher maternal intake increased the odds of typical development in offspring or decreased the risk of offspring having autism spectrum disorder. So again, another positive correlation here, saying that there is no associated risk, that actually the higher supplementation could be a benefit because it could help reduce autism spectrum disorders. In other words, it induces regular typical development in the offspring. So we have now transitioned into the animal studies. So before I talk about this one particularly, I'm going to talk about what differences to keep an eye out for among the animal studies. So main thing is going to be the species of animal use. So some are going to be rats, some will be mice. A big one of importance here is you're going to see that some of these animal studies only look at the male offspring, while others consider both female and male offspring. That's a big one to pay attention to, so try to catch that. But overall, the intervention design is going to be pretty similar across all these animal studies, where you're going to have a control group versus a high folic acid supplement group. And then the offspring's diets may or may not be manipulated. So something to note there, obviously the evaluation measures are going to be different. And then you can again see the overall findings. The most significant ones will be highlighted. But diving into this one in particular, what they were looking at overall was the insulin resistance and its associated risk with high maternal folic acid intake. So if you're looking at those offspring diets and wondering what those abbreviations are, I'll quickly touch on those. Basically, the first initial or first letter is going to signify which diet the mother was on. So C is going to stand for control, F is going to stand for folic acid supplement, and the second letter is going to be the diet that the offspring were on. So again, the C and the F. So some of them flipped, some of them stayed the same, and they evaluated those diets and their effect as well. But overall, what was found is that a high folic acid supplement reduces the availability of adiponectin and promotes insulin resistance in the offspring into adulthood. So if you're looking at that and wondering what the heck adiponectin is, it is a hormone that helps with the regulation of glucose metabolism, and it can actually influence the pathophysiology of insulin resistance. So when we say that there was a reduced level observed in this study, that means that the uptake of glucose by the cells was inhibited, and therefore it did lead to insulin resistance. So that is what this study concluded. So this next animal study was looking at an assortment of evaluation measures, and if you look through some of those, you'll see that they're very similar precursors to the ones that we examine for the risk of metabolic disorder or dysfunction. And that's actually what this study was looking at, is whether or not high maternal folic acid intake is going to predispose the offspring to the development of that dysfunction later on in life. And what the results allude to is that there is a potential that metabolic dysfunction may develop later on in adulthood in the offspring, which is seen in the results through the representation of precursors for this disorder. So also want to note that the study made it very evident that there may be a sex-specific response to perigestational high folic acid supplementation, as all of the significant differences listed here in the results or the overall findings column are pertinent to females only. There were no significant differences seen in the male offspring. So something very interesting to note and to keep in mind for later on in the presentation. What this study was looking at was for behavior modification in the offspring. So overall, what this study found is that high folic acid supplementation in the maternal diet does correlate with dysregulation of gene expression in the offspring's cerebral hemispheres. And ultimately what this means is that it can translate into behavior modification. So with that being said, I will point out the overall finding that's highlighted over here. So the offspring of the mothers who had that high folic acid supplementation, they found that 68 of the genes were downregulated and 56 of the genes of the offspring were upregulated. So that goes back to that dysregulation. And one gene that I want to point out in particular that they noted was a gene labeled HTR4, and this is a gene that's associated with autism. So there is another study that was looking at autism that we talked about earlier, which actually said that there was not an associated risk for an autism spectrum disorder with high folic acid intake, whereas this could kind of turn into that. So it could translate into an associated risk, but we'll touch on that later. This study was focusing on the potential risk of seizure disorder development in the offspring exposed to high folic acid intake through the maternal diet, and how they did this was the measurement of the seizure onset time when the offspring were subjected to a high dose of canic acid. So canic acid is known to induce a seizure, but they were just seeing just how quickly that was going to occur and if it had an association with the maternal folic acid intake. So also they were looking at the firing rate of neurons of some tissue samples that were grown in vitro, and I will note a little bit more on what that means in a second, but overall what the results of this study point to is the fact that while high folic acid supplementation alone might not be sufficient to result in a seizure disorder, it could contribute to the possibility, but only in combination with other factors. So when they were looking at that firing rate, they did see that folic acid supplementation did cause the neurons to fire bursts rapidly, and this was not observed in the controls. So what this rapid firing rate translates into is an increase in neuronal activity, and that is then translated into accelerated development of neuronal connectivity, and in turn this leads to a hyperexcitable network. So basically saying the hyperexcitable network just means that they are more prone to seizures and that the neurons are just going to react far quicker. And so overall, yes, the seizure onset time was shorter for the offspring subjected to folic acid supplementation through the maternal diet. So this is our final study that we will be looking at, and it again was looking at the insulin resistance in offspring. And one thing to note in this study is that they had a control group in the high folic acid group. They also included a regular group, and their justification for this is the fact that the regular group, that 5 milligrams of folic acid, that amount in mice is going to translate to the recommended amount for humans, which is that 400 micrograms per day. So that was an interesting change in the intervention design as far as this study goes. But overall, what they found is that maternal high folic acid supplementation, but not regular supplementation, exacerbated the effects of high fat diets on glucose tolerance and insulin resistance. So I forgot to mention that the offspring were subjected to a high-fat diet. The reasoning behind that being that here in the U.S., in our westernized culture, we follow that westernized diet, which has been associated with a high-fat intake. So we're just trying to see the correlation of all three things, kind of trying to mimic what we see in humans, but they did it on animals. So the adiponectin was again an evaluation measure for this study. So what they found is that the adiponectin expression in the mRNA was altered, and they said that this was likely caused during the intrauterine exposure to maternal folic acid supplementation, and not so much in correlation to the offspring's high-fat diet. And the reason they say this is that there was a consistent decrease in adiponectin mRNA levels of the high folic acid offspring group, and the low serum levels of adiponectin, were seen before the implementation of the high-fat diet. So overall, there was the associated risk of glucose intolerance, especially due to that reduction in adiponectin. So to wrap up these studies, I have put their overall findings into this little table here. So on the left side, we see the positive outcomes associated with high maternal folic acid intake, and on the right side, we see the negative outcomes. So then the two listed here in red, so side to side, they both were evaluating the same thing but had a different result. So starting with that first row there, that discrepancy is most likely due to the evaluation measures used. So the study by Trivedi mentioned that spirometry is more of an accurate indicator of asthma if the subject is currently undergoing an asthma attack or has a high prevalence of the symptoms at that time. Whereas the study by Parr only utilized participants whose offspring currently had an ongoing asthma diagnosis. Therefore, both studies had some limitations to them within themselves, so it's hard to decipher which one is more accurate. So it's hard to draw a conclusion on the associated risk with asthma. And then that second row there, the study by Barua on the right side mentioned that the HTR4 gene that was dysregulated by a high folic acid exposure, it was mentioned as having a correlation with autism, so that's why I put these two next to each other. So there was dysregulation of gene expression, which could lead to a behavioral modification as seen in autism spectrum disorder. But again, can't really draw a fine conclusion based on just those two studies. We just need more human studies in general, but give this table a look over as these are the overall results from this systematic review. When looking at other systematic reviews that were examining the same kind of topic as I was, I noticed that many of them also found conflicting results as far as the autism correlation goes. One thing that the study mentioned is that folinic acid has been observed as a better source of folate for the prevention of autism. And what folinic acid is it's a form of folate that's actually able to overcome genetic mutations that affect proper folate metabolic pathways. So we know there's that MTHFR genetic mutation that can impact the metabolic pathway of folate getting in and utilized in our systems. So what they're saying is that they found that folinic acid might be a better source of folate, and that it might have a better overall impact on reducing the risk of autism. Another review pointed out that there is conclusive evidence from animal studies, but more human studies are definitely needed to determine appropriate dosages and timing of folic acid intake. So what they found in their animal studies, which they said were conclusive, was that behavioral, morphological, and molecular changes were seen in the brains of the offspring. So very similar to what this systematic review pointed out. A lot of those animal studies did point to all those things, but the human studies, there were conflicting results, and we just can't really draw from that. We definitely need more human studies. I wanted to briefly discuss the current folic acid regulations and recommendations. So right now, the World Health Organization recommends women take a folic acid supplement of 400 micrograms per day from the time they plan pregnancy until 12 weeks of gestation. So there is an emphasis on the time they plan pregnancy. As we've seen in some of the studies in the systematic review, the timing of the folic acid supplementation can definitely play into the results of the offspring's health. So that's important to note. But again, this just goes back to this is the amount that has been found to be effective in reducing neural tube defects. So I just wanted to include here the top selling prenatal vitamins and their amounts of folic acid. So I have the top 5 listed. These are from Amazon's best-selling list. And the one that's bolded there was surprising to me because it is far above that tolerable upper intake level that has been put into place by the Institute of Medicine, which is 1,000 micrograms of folic acid. So this is very interesting to me, but you can see that overall all of them are above that 400 recommendation, and we have to consider the fact that this is not the only source of folic acid that mothers might be getting in through supplementation, because they also are eating diets that are fortified with folic acid, and there are many natural food sources of folate as well. So as far as the limitations go for the systematic review, the dietary intakes of folic acid could definitely vary based on country. So we saw that in the human studies, some countries had no mandatory fortification practices while some did, and that did we could see that impacted the overall mean folic acid supplementation in the mother's diets and supplementation use as well. So anyways, that definitely was going to vary. And then also the dietary intakes of folic acid can vary amongst individuals even in the same country. And this is just going to go back to that everyone eats their own diets, everyone has their own preferences, so even if they're in the same country, we can't ensure that they're getting in the same amounts of folic acid through their foods. Again, this has been pointed out several times in this review that animal studies are just hard to project on humans. We definitely need more human studies and more concrete results from them. With that being said, there are just so many confounding variables to adjust for in human studies. I mentioned earlier that in animal studies we can really control those extraneous variables, whereas in human studies it just gets harder and harder to do that, as humans are very individualized and there's so many other extraneous variables that can play into the results. And one thing too that I had hinted at was the fact that the animal studies, some of them only looked at male offspring, and some of them looked at both males and females. So the reasoning behind some of the studies only including male offspring is the fact that it has been found that female rat hormones can affect study results. So that's why they wanted to throw that extraneous variable out and only look at males. So that definitely was another limitation, the fact that some of them were consistent with controlling for that variable and some of them were not. Transitioning into the implications and conclusions. So some implications for future research. The main 3 things here are looking at the timing of folic acid supplementation. So at what points in the pregnancy is it most important? Also looking at the fact that unmetabolized folic acid has been seen in mothers and offspring, and also what other nutrients are of concern besides folic acid in the maternal diet, and what implications can they have on the offspring's health. So starting with the timing, we know that the neural tube closes at day 27 of human gestation. So after this point, researchers are starting to question if high levels of folic acid after this neural tube closes is even necessary. So again, just looking at what point in the pregnancy is it most important, and maybe is there a time at which that supplementation can stop or slow down. And then excess unmetabolized folic acid has been seen. It's been revealed in studies that show that the amount of dietary folate intake is far less than what's observed in the serum and red blood cell folate concentrations. So this is seen not only in mothers, but also this evidence of unmetabolized folic acid has been seen in the fetal cord blood. And so there's just questions being raised as to what does this unmetabolized folic acid mean? What implications could it have? And then we also know that detrimental effects on offspring's health have been observed later in their life due to poor nutritional quality of the maternal diet. So again, that just goes back to what other nutrients do we need to be concerned about with the maternal intake and its implications in the offspring's health. So to conclude the systematic review, I will say that as future dietitians, the best thing we can do is to make sure that mothers or pregnant women know the current recommended level of folic acid intake and where that is going to come from in both their diet and from their supplements that they might use. And overall, just how to choose a good supplement and how to read those labels to know how much folic acid they're getting from them. So it just goes back to education, making sure that we are educating our patients and clients and that they are aware of these things. Especially want to make them aware of the tolerable upper level intake that has been set for a reason, and that they should approach it with caution. So it definitely is important for mothers to take in folic acid in the early weeks of pregnancy. That has been well established. But as far as taking the concerns of that high folic acid intake to an extreme by changing the recommendations, I don't think we are there yet. We definitely need more human studies to draw some more concrete conclusions. But for now, again, just educating and making sure people are aware of that range of the 400 to 1,000 and how they can kind of monitor their intake through not only their supplement use, but also what they're getting in through their diet. I will now leave you with my long list of references on the next 3 slides. And I just wanted to say thank you for watching this presentation, and I look forward to seeing what questions you guys have, and I hope that I can answer them to the best of my ability. Thanks, guys.